Bismillahirrahmanirrahim. Assalamu alaikum. Our today's topic is about stages of Meiosis 1 division. Lecture number 10, class 9, chapter 5, cell cycle. Division 1 of meiosis. This is the meiotic division in which the number of chromosomes is reduced to half, and from one nucleus, two nuclei are formed. It consists of following stages. Prophase 1. This stage consists of further 5 stages, that is leptotene, zygotene, pectine, diplotene and dikinesis. Leptotene. In this stage, chromosomes appear in the nucleus. Each chromosome has an identical partner. One chromosome comes from the male and one comes from the female. They are called homologous chromosomes. During this stage, the chromosome network breaks into a specific number of long, thin, beaded thread-like structure called leptotene. Each thread has two morphological similar leptine in each cell called homologous structure. Zygotene. During this stage, the homologous chromosomes, which are similar in structure, which comes from the mother by ovum and father by sperm, are attracted towards each other, and their lengthwise pairing takes place. The pairing of homologous chromosomes known as synapses, while the paired homologous are known as bivalent. Pectine. These bivalent chromosomes become shorter and thicker due to their increase in coiling. Then each homologous chromosome divides into two chromatids. In this way, four chromatids are formed. They are called tetrad, consist of four homologous chromatids. The synaptic forces of attraction between each bivalent decrease and the chromosomes uncoil and separate. The separation is, however, incomplete and paired chromosomes are in contact with each other at one or more point called chiasmata. Each homologous chromosome splits longitudinally except in the centromere region. Now, each bivalent is composed of four chromatids and therefore is known as bivalent tetrad. Diplotene. The homologous chromosomes are attached in one or more point called chiasmata. At the chiasmata, an exchange of genetic material occurs by a special process called crossing over. The chromosomes are coiled in a thick shield of matrix. Dikinesis. The chromosomes are almost separated from each other except at the chiasmata. The nucleolus disappears and nuclear membrane also starts to degenerate. The chiasmata moves towards the end of chromosomes. This process is known as terminalization. Nuclear spindle begins to be formed. Now metaphase 1. The bivalent line up at the equatorial plane. The centromere of each chromosome attaches with same fibers of spindle. Anaphase 1. At this stage, one chromosome from each member of homologous pair begins to separate and move towards its respective pole by the contraction of spindle fiber. Telophase 1. The nuclear membrane form around the chromosome at each pole and chromosomes become uncoiled, the nucleolus reappear and the two daughter nuclei formed. Cytokinesis. Telophase may or not be accompanied by cytokinesis and daughter cells formation. Quote of the day. The aim of education is the knowledge, not of facts but of values. Thank you very much. Please like, share, and subscribe my channel Science Kick. Allah Hafiz.